So today in class, we started discussing what life is, and we ended here on the 7 characteristics of life. So on the test, I will not ask you to list the 7 characteristics of life, but you should be aware of what they are and be able to recognize them. So in a multiple choice question, I could say which isn't or which is a characteristic of life, but I won't ask you to list them. The 7 characteristics of life are, they are composed of cells. All living things are composed of cells. It's part of cell theory. Organization. Cells are highly organized. We're going to be spending a majority of the class discussing how the cell is organized and how highly regulated it is in order to sustain life. Living things grow and develop. A rock is a rock. It does not grow. It does not develop. Everything that is alive grows and develops. Yes, even bacteria grow and develop. Things that are alive use energy and have a metabolism, and regulation and homeostasis. So what is metabolism? Metabolism is the sum of all of the chemical reactions in a cell. So we talk about having a fast or slow metabolism, but that's talking about how we break food down. And we'll get into more detail about the different types of metabolism a little bit later. Homeostasis means a steady state. It's the body's ability to maintain a constant internal environment. For example, the pH of your blood is about 7.3-ish, and it maintains that pH. Anything higher or lower causes reactions to occur that will bring the pH back to what it's supposed to be. Another example of homeostasis is temperature. So it's going to be like -20 degrees tomorrow, and if you walk outside, you'll get cold pretty quick, but if you're only outside for a short amount of time and take your temperature will still read 98.6 degrees or thereabouts, because your body will increase its metabolism in order to generate heat to keep your body temperature stable. Number 5, things that are alive respond to environmental changes, to stimuli. Yes, even the lowly prokaryotes respond to stimuli. If you put something in a microneedle and put it near a bacteria, if the bacteria likes what it senses from the needle, it will move towards it. It will respond to stimuli. If it doesn't like it, it will move away. Number 6, reproduction. Genes are passed down from generation to generation. I'll talk a little bit more about that in a second. And number 7, biological evolution. If you want to know more about this, you should look into Biology 111, which is ecology and evolution. I will briefly touch on this in a few slides, but for now, we'll leave it at that. Organisms also have several levels of organization. Atoms and molecules form cells. We'll be talking about atoms and molecules in the coming lecture. These cells then associate to form tissues. Tissues organize into functional structures, which are your organs. So liver tissues make up your liver, bone tissue makes up your bones. Then you have different organs that come together to make an organ system. So for example, the digestive system, you have your mouth, your esophagus, your stomach, large, small intestine. All of this make up an organ system. And then together, all of the organ systems make up an organism. So the most basic level of organization is the atom. And then atoms join to form molecules, which form cells, and eventually we end up with the organism. So those 7 characteristics of life unify life. All life displays those 7 characteristics, from the lowly prokaryote all the way up to the mighty redwood. All share the same characteristics of life. However, there's a large difference between the prokaryotes and a redwood, between us and a fern. So diverse forms of life form in diverse environments. So life began on Earth as primitive cells about 3.5 to 4 billion years ago. These primitive cells underwent changes that gave rise to the species that we see today. And understanding this helps us understand the structure and function of an organism's body. So evolution is one of the most important unifying concepts of biology. So we're not going to spend much time on it, but I wanted to give you a brief overview of what it's all about. Evolution is the process by which populations of organisms change over time. These are populations, not individuals. We are not evolving. We are done. But the human population as a whole is evolving, but not individual people. Evolution involves passing genes for new traits from one generation to another, leading to differences in populations. There's 2 mechanisms of evolutionary change. The first is vertical descent with modification. This was Darwin's theory. You might be familiar with the saying survival of the fittest. It's called vertical descent because if you picture a family tree, you start at the top, you have your grandparents, and then as you move down the tree, you have your parents, and then you, and then your children. So that's the vertical part. The modification occurs when the sperm and the egg unite to form the new organism. There's different modifications that can occur, and we'll talk about those in the last unit of the class. But these different modifications over time, over thousands of generations, can form a new species. Natural selection can take advantage of beneficial mutations. Natural selection essentially means that some mutations allow one organism to have an advantage over the other. So that mutation is selected for in that that organism will have offspring. So there were two competing theories in the mid to late 1800s. We have Lamarck and Darwin. So what Lamarck believed was that acquired traits were heritable. So things that we did or that we learned or that happened to us during our lifetime could be passed on to our offspring. So today we know, well, that's kind of ridiculous. I mean, if you dye your hair purple, your offspring won't have purple hair. And if for some reason someone lost a limb, their child would not be born with a missing limb. However, science has come to learn that some acquired traits may be heritable in the form of epistasis. So if you're interested in that kind of discussion, you might want to look into taking our genetics class because that is a very complicated method that our DNA has of changing itself under different conditions. So let's take a look at this diagram. We have some giraffes here. So according to the theory of Lamarck, the giraffes had short necks and the trees were too high. The leaves of the trees were too high. So in order for the giraffes to reach the leaves, they had to stretch their necks, causing their necks to elongate. Therefore, in the next generation, those giraffes would have longer necks because their parents stretched their necks. Again, we see this as ridiculous. Well, that's silly. Of course not. That doesn't happen. Darwin had a different theory. Darwin's theory is a little bit sadder, but what he found, what he theorized, is that you have two giraffes. Due to genetic variations in the population, one giraffe happens to have a shorter neck and one giraffe happens to have the longer neck. The giraffe with the shorter neck can't reach the leaves. The giraffe with the longer neck can. So now the giraffe with the longer neck has lots of food to eat. He's big and strong. A potential mate shows up. The mate is likely to choose the big, strong giraffe. Even if the smaller giraffe decided to put up a fight, it's unlikely he could defeat the larger giraffe in a battle for the mate. So the giraffe with the longer neck would get the mate and therefore leave offspring. The shorter neck giraffe dies, no offspring are left behind. So that gene for the short neck would eventually disappear because the shorter neck giraffes weren't finding the mates. And this same process continued over many generations. The giraffes with the longest necks were the ones that survived. This is natural selection. Nature selected for the giraffes with the longer necks because they could reach the leaves, therefore they could have offspring. The second type of evolution is called horizontal gene transfer. This is genetic exchange between different species. This is relatively rare and it only happens in prokaryotes. So the genes that confer antibiotic resistance are a wonderful example of horizontal gene transfer. So here's an example. We have a bacteria species such as E. coli, and there's an antibiotic resistance gene located here. What the E. coli will do is send out essentially a spear and stab another bacteria with its spear. And usually it's of a different species. Once this spear has landed into the new bacteria, this antibiotic resistance gene is copied and it is shared with the new bacteria, and now this strep bacteria now contains the antibiotic resistance that previously was found in the E. coli. This happens a lot in hospitals when you have a lot of sick people coming together and a lot of different bacteria are mixing. It is very rare. It seems like it happens more often than it does, but being stabbed with a spear is not really great for the strep. So oftentimes this bacteria will not survive the spear throwing of the E. coli, and other bacteria do this as well. It's not just E. coli. But when it does, now we have a strep that has an antibiotic resistance gene. And this is likely often happening in our guts as well, because that's also where a lot of bacteria can mix. So vertical descent with modification happens over generations among a single species, whereas horizontal gene transfer happens in the same generation from two different species of prokaryotes. Right. One of the major themes of biology that we're going to be talking about this semester is the difference between the genome and the proteome. So the genome is the complete genetic makeup of an organism. It's essentially the DNA. So your genome is your DNA. Your DNA is the same in every cell of your body. The two exceptions are your germ cells, sperm and egg, and your blood cells, which don't have any DNA because they lack a nucleus. But other than those two exceptions, the DNA in every other cell in your body is exactly the same. So we can look at the DNA and use our DNA to determine evolutionary history from looking at the genome. I'm sure many of you have heard of the Human Genome Project. That has been done. There have been thousands and thousands of DNA sequenced. People's DNA sequenced. You can order a kit from 23andMe or Ancestry.com and have your own DNA sequenced, at least parts of it. They don't actually sequence your entire genome. But we have a pretty good hold on a lot of DNA sequences. We don't know what they do, but we know what they are and where the differences are in between individuals. Proteomes are a little bit more complicated. So, for the proteome, you need to know the central dogma of biology. And the central dogma of biology says that we start with DNA, and DNA is used to create RNA, and then RNA is used to make protein. Pardon my horrible writing with the mouse. Now, if you don't know what DNA or RNA is and you don't understand what protein is, that's fine. We're going to be talking a lot about that in Chapter 3. For right now, what you need to know is that we start with DNA, and DNA codes for protein with RNA as an intermediate. Proteins are the functional units of the cell. Everything that the cell does is done by a protein. So proteins are highly important to the cell. That's what makes the cell run, and we get our proteins from our DNA. So as I mentioned in the previous slide, the DNA is the same in all cells in an organism. So your eye cell has the same DNA as your skin cell. They are exactly the same. But your eye cell and your skin cell are obviously different, and that's your proteome. So the proteins that your eye cell makes are different than the proteins that your skin cell makes. So your eye cell makes proteins that give your eyes color. Making that in a skin cell would be ridiculous. You don't want your skin being green or blue or purple, so that wouldn't work out very well, and it's a waste of energy for the cell to make a protein that it doesn't need. So the proteome is the different proteins that each cell can make. Your liver cell makes a protein that helps create bile for your liver. You certainly don't want the bile protein active in your eye. So your eye makes different proteins than your liver, which makes different proteins than your skin. And all of the proteins that one cell makes, we call the proteome. And this is a theme that we're going to come back to throughout the course. All right, so an example of this genome versus proteome is the crystal jelly. This crystal jelly is bioluminescent. It makes a green fluorescent protein conveniently called GFP for green fluorescent protein. The gene for GFP is in the jellyfish genome. Every cell in the jellyfish has the ability, it has the gene to make GFP. But the GFP is only expressed, that means the protein is only made. When we say a gene is expressed, that means it's making a functional product. The GFP is only expressed in the proteome of cells that form glowing spots. So here we have the jellyfish. You can see here we have the glowing spots around the edge of the jellyfish creating the GFP protein. Here in the hood of the jellyfish at the top, we don't see any GFP because those cells are not making the protein. So even though they have the DNA, the same DNA as these cells, the proteins that each cell makes are different. And we're going to be talking about this throughout the course. All right, so a little bit of vocabulary because it's generally found that people confuse hypothesis and theory. You might have heard recently all this stuff about a flat earth theory. Flat earth is not a theory. We'll talk about what a theory is in a couple minutes. Let's start with a hypothesis. A hypothesis is a proposed explanation for a natural phenomena based on observations. So it's not just something that we pull out of a hat. It is not a guess. Scientists do not guess. Usually when we have a hypothesis, we're pretty sure it's going to work. Although not always. Sometimes it doesn't work. Sometimes we're just taking a stab at something. But it's not a guess. It's based on observation. So for example, maple trees drop their leaves in autumn because of shortened amount of sunlight. So what was the observation? Someone said, hey, it's autumn and the maple trees have dropped their leaves. So why would they drop their leaves in autumn? Well, maybe it's because there's a shorter amount of sunlight. There are other hypotheses too. Maybe it's the age. Maybe it's the amount of wind in autumn. It could be a lot of things, but based on observation, the hypothesis is it's because of the shortened amount of sunlight. And we're going to come back to this hypothesis when we talk about the scientific method in a few minutes. So hypotheses must make predictions that can be shown to be correct or incorrect. That means it has to be testable. You can't come up with a hypothesis that you can't be proven wrong about. So we see all these shows on the Discovery Channel. Bigfoot exists. That's their hypothesis. But you can't prove that wrong. It's impossible because when they don't find Bigfoot, what do they say? Well, Bigfoot is hiding. Bigfoot is shy. Bigfoot saw us coming and he ran away. Bigfoot doesn't like the smell of people. All these excuses for why they can't find Bigfoot. So that's not a testable hypothesis because no matter what happens, they will never be proven wrong. A better hypothesis would be there is no Bigfoot and then go out and look for it. And then if you find Bigfoot, then you can be proven wrong. A hypothesis is never really proven. We never say the word prove in science. We may never have found the true explanation for a phenomenon. And I'll talk a little bit more about that when we get to the theory. So a theory is a broad explanation of some aspect of the natural world. It allows us to make predictions. Also, it can never be proven true. But due to overwhelming evidence, it's extremely likely to be true. So back to the flat earth. You can't say flat earth is a theory because it is not substantiated by a large body of evidence. It is substantiated by zero evidence. So it is not a theory. It is at most an idea. That's not true. We can't make predictions because any predictions that we make would be wrong. So for example, the theory that DNA is the genetic material, an overwhelming body of evidence supports this theory. However, prior to the 1930s, there was a theory that protein was the genetic material, that protein was what caused heredity, and there was a lot of evidence that supported that theory. It wasn't until the late 1920s, early 1930s that people started looking at DNA as the genetic material. And once again, scientists were fighting because when it was shown that it was not protein, that it was DNA, people had a really hard time letting go of the protein as the genetic material theory. But when looking at the data from the proteins being the genetic material, all of that data actually made sense if DNA is the genetic material as well, because back when that data was found, we didn't know much about DNA. So all of that data for protein is actually data for the DNA being the genetic material. So that theory was actually disproven. We never say anything in science is proven. We say it's supported and it's probably true, but we can never say it's proven because new data and new techniques could show that it's not true. So the two key attributes of a theory: consistency with vast amounts of known data and the ability to make correct predictions. So can a scientific theory be rejected? The answer is yes. New experimentation can lead to data that does not support the theory. So yes. So the scientific method, I'm going to very briefly go over this. So there's 5 stages. Once again, I'm not going to ask you to list the 5 stages. You need to be aware of what they are and be able to identify them, but I'm not going to ask you to list them. So first, observations are made regarding natural phenomena. Oh, hey, look, it's autumn and the leaves are falling from the trees. These observations lead to a hypothesis that tries to explain the phenomena. Hey, it's autumn, the leaves are falling from the trees. Perhaps it's because of the shortened amount of sunlight. Then an experiment is conducted, which I'll show you in the next slide, to determine if the predictions are correct. Then we have to do number 4. We have to analyze the data. So a lot of times in lab, students forget that part. They get all the way up to number 3, they do the experiment, and they think they're done. But you're not. You actually have to analyze your data and then accept or reject your hypothesis based on your data. Your data has to be explained, usually in some sort of a table or a graph. All right, so observation. Leaves on maple trees fall in autumn when the days get colder and shorter. Hypothesis, the shorter amount of daylight causes the leaves to fall. So here's our experiment. Small maple trees are grown in 2 greenhouses where the only variable is the length of light exposure, the amount of time. So here we have this greenhouse where the light remains constant for 200 days. So for example, 12 hours a day for 200 days. Here, the amount of daily light becomes progressively shorter for 200 days. So maybe it starts at 14 hours, and then it goes down to 10, and then 11, and then 8, and then 6. This group is called the control group. The control group is very important because the control group is unchanged in the variable. So we didn't change the variable being the amount of daylight. So that's constant. You need this control group so you can compare it to the experimental group. So let's look at the data analysis, analyzing the data. The control group only lost maybe 20 leaves, but the experimental group lost 200 leaves. So by comparing the control to the experiment, we can say by changing the amount of sunlight, more leaves fall. If we didn't have the control group, if this was gone, not here, then we'd have nothing to compare it to. Okay, so 200 leaves fell. Big deal. What does that tell us? It wouldn't tell us anything because maybe in another group 400 leaves fell or 50 or 30. We have no idea. So by using the control group, we have something to compare the experimental group to. So we have the data. You'd have to do some statistical analysis, get some error bars on here. We're not going to really be doing too much of that. We have one statistics test that we're going to use in genetics. And then you make your conclusion. The hypothesis is accepted because by shortening the amount of daylight, more leaves fell. All right, I think that about wraps it up. I tried to keep this pretty short, not too bad, and I will see you in class.